all living things, regardless if they are a single-celled amoeba or bacteria, or a complex polar bear, complete the following 6 criteria. So why aren't viruses alive? Well, for one, they're not even cellular. Do viruses eat and excrete? No. You can leave a virus in a test tube or on a petri dish indefinitely. And assuming there's no damage to the virus structure, it could still be infectious. Do viruses grow and develop? No. That genetics and protein shell does not change going in, going out, or over time. It just sits in stasis unless your cells or the host organism cells do something about it. And last but not least, viruses don't reproduce on their own. They lack the machinery. You put 2 virus particles in a petri dish and leave it with all the food and resources all the time in the world and come back, you will still have only 2 virus particles. On their own, they do not have the mechanics to generate more. So let's compare and contrast these 3 non-living things. A virus is genetic material in a protein shell. That protein shell is called a capsid. Okay, this is different than the plasma membrane or the cell wall. It's important to note that because of their size, viruses either have DNA or RNA. It's one or the other. They can't have both. There's not enough room for it. So you either have a DNA virus or an RNA virus. So just one set of instructions and a protein shell. In that sense, viruses are sometimes called genes in a suitcase. Or I like to think of them as the plain M&Ms, right? The chocolate gooey center is the genetic material. That's the part that has value and worth. And the protein capsid is that sugar coating that keeps it from melting. So you have a delivery mechanism into your mouth. A viroid is smaller than a virus. And what separates it is that there is no protein shell. There's no capsid. A viroid is always just naked, infectious RNA. And then there's the prion. A prion is a very specific type of infectious protein. It's important to note here that proteins are different than nucleotides and nucleic acid. Instead of A, T, C's, and G's or A, U, C's, and G's, it's being composed of amino acids. It's important to point out that not all viruses are bad. The big thing here is we want to consider host range. Okay, this is the distribution or the variation that exists in which organisms can be infected by a virus. It's the variety of living things a single virus or bacteria could infect, because host range is a term that's not limited to viruses. This is determined by the genetics of that organism, as well as cellular and host factors. Okay, that partnership, that relationship. For viruses, because of their limited genome size, typically their host range is very, very narrow. Not only can they only infect one type of organism, they generally can only infect one type of cell inside that host. We refer to this as specificity. It's a one-trick pony. Now, there are exceptions to these rules. Flu infects a large number of organisms. Rabies infects a large number of organisms. But on the whole, the viral host range is pretty limited. And so there are a number of viruses that do not concern humans because we are not their definitive host. Most viruses are self-limiting diseases, meaning they're going to run their course and resolve spontaneously on their own, with or without specific treatment on your part. And that's because the virus life cycle typically kills off their host cell and they burn themselves out. There are those viruses, though, who are potentially latent, that they infect long-lived cells that don't rupture, and they can remain asymptomatic in the host organism for a long time. It's basically a hibernation period or a period of dormancy. During this time, the person is infected, but not diseased. The virus is present, but not transmittable, not contagious. Functionality of the organism is normal. A good example of a latent virus, herpes simplex 1, the causative agent of cold sores. Once infected, you are infected for life. But the reason why we call them cold sores is because we typically only see those symptoms appear when your immune system is otherwise taxed by something else. And it's during those times that the immune system cannot hold the virus at bay, and so we see a short amount of viral replication. The disease runs its course, you get an ugly blister for a week, and then it goes away. The virus is still present, but now it's held in a state of dormancy. Another example of this is chickenpox. Once infected, you are infected for life. The chickenpox virus lives in our cells, but generally speaking, we do not exhibit chickenpox symptoms because our normal healthy immune system is able to hold those cells at bay and prevent them from replicating or producing their genetic components. However, as a person ages, their immune cells diminish. And so usually around 55, 60s, there's less immune cells present. We consider the person immune compromised, and those cells that were infected with the chickenpox virus can rear their ugly heads again, and you see chickenpox reoccur. This time we call it shingles, and the reason for that is because the manifestation of that disease is different. Instead of spotted, itchy rash, these are lightning strikes of heat and pain, and the reason for that different presentation is because the biochemistry, the body chemistry has changed in the organism, the human host, because of differences in diet, age, the makeup of types of cells that exist in the body. Okay, the virus has always been there. It was just latent during that time. And then last but not least, persistent viruses. Persistent viruses are the name that we generally give to RNA viruses because these are viruses that are infectious and pathogenic from the get-go. However, they progress so slowly that generally the disease aspect is unnoticed until it's almost too late. A good example of this is the measles virus, which has a mortality rate of 25%. So you have a 1 in 4 chance, if you had measles, of dying. And the reason for that is because it's not just a rash on the body, but the measles virus is small enough to cross the blood-brain barrier. And in 25% of individuals infected, it causes subacute sclerosing panencephalitis, which is just a fancy way of saying massive brain swelling. In this situation, the first sign that you have subacute sclerosing panencephalitis is spontaneous death. Too little, too late to know or do anything about it. How can we prevent viral, viroid, or prion diseases? A lot of it has to do with good hygiene. Avoiding contact with contaminated food, fluids, washing our hands frequently. Even though these organisms are not alive, disinfecting surfaces can remove them from the environment and proximity where you would otherwise acquire them. The idea here is a vaccine is made up of a weakened or killed component of the pathogen, whatever that may be, and it's meant to trigger your body's natural memory response, the natural immune response of the body. In these panels, what we're seeing is we've introduced a small dead virus, we'll say flu, for example. It cannot cause flu, it cannot actually make you sick, but now your immune cells, depicted in the no, find a way to kill it off, to destroy it, to remove it from the cell, to clean house. It resolves this problem on its own. But those immune cells are long-lived, and so they will hang out. So when you encounter the real threat in nature, they are already present and ready and able to respond. Vaccination is not possible without our natural immune response that we currently have. If you otherwise get a viral infection, all we can do is supportive care. We can try to make sure that you're comfortable, keep you hydrated. Supportive care works really well for self-limiting viruses, just managing the moment until the virus goes away on its own, when the cells die on their own and take the virus with them. But in terms of those very pathogenic scenarios, supportive care is really just us hoping for the best. Some viruses have the ability to have antiviral medication provided. This is not a Band-Aid for all. It is not a regular treatment option that we have available. It is available for only a few specific viruses. And what antivirals are is they identify a very specific target of the viral life cycle, and the goal there is to inhibit that step. So antivirals are useful in slowing down virus's development or alleviating a symptom. But in this case, it basically falls under the category of supportive care, hoping that your body, your immune system, can do much of the legwork in removing it. An example here is HIV has a number of antivirals available to it. Some antivirals target the attachment of the HIV virus to the T cell. Some antivirals target the replication machinery so that the HIV virus has integrated into the host cell, but it cannot be copied. Some antivirals target protein processing, which is the ability of the virus and the viral genome coming together. And then some of the antivirals target virus maturation, which is a fancy way of saying the virus leaving the cell and going on to infect someone else. And so this way you might have a cell that's so full of viruses, but they can't seem to leave in one way or another. The point that I want to make here is antivirals are not a cure. The virus is still present. There's harm still being done here. Well, the type of nucleic acid, whether it's DNA or RNA, makes a big difference because it affects speed. The general flow of genetic information is for DNA to be made into more DNA in a process called replication. Then the DNA can be transcribed into RNA and leave the nucleus. And that RNA can then be translated into proteins that have functionality. If a virus is a DNA virus, it is already speaking the same language. To incorporate into the host genomic structure, all the virus has to do is approach, attach, insert its genetic material, and it can assimilate, like to like. So there's only really 4 steps here. The RNA virus, on the other hand, is speaking a different language. It must be reverse transcribed from RNA back into DNA before it can insert. So this creates a new step. The virus can approach, attach, insert, but then reverse transcribe prior to insertion. And so that added step creates a slower progressing disease. Retroviruses, which is what we call RNA viruses, because retro means to go backwards, are slower diseases. The classic example of this is HIV. HIV is such a slow progressing disease that 1 in 4 people are infected but have no idea, because outwardly they're still functionally healthy. The type of protein coat can be in a wide variety. They can be tubular and hollow. It could be a polyhedral, 3-dimensional, or it could be complex, like the bacteriophage here on the right. Shape of the capsid will determine where it attaches and how it enters. That's its significance. At its core, the capsid is just a glorified delivery system. The last way we can classify viruses is whether or not they have the capacity to obtain an envelope. The virus does not come stock with an envelope. This is something that some viruses have evolved to acquire from their host. And we'll talk more about the viral life cycle strategy in a few slides, but we can see in this example here, this particular virus has the genetic material in red, the capsid in green, and surrounding that is a plasma membrane stolen from the host as it left. And that's what we call an envelope. And the value of being able to do this is that now that virus is a wolf in sheep's clothing. It has the capacity to hide for a time from the immune system, because at first glance, any immune cell will see it and say, oh, those are my plasma membrane cells. As we move forward, let's talk about replication strategies. Viruses are obligate parasites because they cannot exist on their own. They are often called metabolic pirates because they take over your cell. They use your cell's resources. They use your cell's building blocks and your cell's machinery to achieve their end game. Right? So panel 1, the virus is going to approach a susceptible cell. Assuming it's the right type of cell that the virus can infect, it will attach to that cell using specific proteins embedded in its capsid. From there, it will deliver the genome. Either that means it absorbed in and can take the genome to the nucleus, or it just injects the genome in. And then we've got panel 4, which we saw on a previous slide. That DNA, or the RNA reverse transcribed back into DNA, will get integrated into the host genome. From there, there are 2 potential outcomes, 2 types of growth cycles that we've described in detail, seen in viruses, bacteriophage viruses that infect bacteria, but it has been confirmed to occur in human viruses as well. Know the difference between the 2 growth cycles. Be able to recognize the unique steps that take place here, but also the implications of this type of infection. The first one we will discuss is the lytic cycle. Okay, so up through now, steps 1 through 4 are consistent. But now in the lytic cycle, the virus has hijacked the host cell machinery. And so when the host cell's ribosomes read mRNA. It's building host proteins and virus proteins. When the host cell copies the host genome, it is also copying the bit of virus DNA that's injected inside too. Then in panel 6, the virus particles self-assemble. They come together on their own. How are they able to do this? It really goes back to chemical atom affinities, what the molecules like and how they're going to bond. And so there's an attraction between the viral capsids and the viral genomes that help them find each other in the cellular space and absorb the correct material. I like to think of it in terms of visualizing, like the Terminator T-1000 from Terminator 2. He was made out of liquid mercury, and when John Connor shoots him and Arnold Schwarzenegger does some damage, all those bits and fragments of him pull together and reconstitute him. That's kind of how these virus particles work. Okay? There's much more steps involved, and at its core, it's the atomic particles driving this. And then that takes us to the final step in the lytic cycle, where so many of these virus particles have been made that the cell becomes chock full and ruptures. That type of damage will result in the death of that particular host cell, and all those virus particles are now released and infectious from the moment they exit and can go on to infect a neighboring cell or other cells nearby. This is in contrast to the lysogenic cycle. In the lysogenic cycle, there's a pause after the shared steps 1 through 4. And the virus essentially lays dormant. But whenever the host cell is duplicated during mitotic division, the viral genome is also duplicated. It lays there like a sleeper agent. The lysogenic cycle is sometimes referred to as a temperate virus life cycle because in this case, the host cell is not damaged. The host cell is not killed. The danger herein lies the fact that when these viruses insert, they are disrupting the flow of the host cell's genome, and therefore can disrupt the normal functionality of that cell. In fact, 11% of all human cancers have been traced back and tied to viral genome insertion. Now, I want to take some time applying what we know about viruses to one particular case example. Flu belongs to a family called Orthomyxoviridae. This is Greek for mucus. So this attributes the fact that very early on, we recognized that this was a respiratory disease. In terms of flu, there's not just one flu. There are 3 different types of flu, and within these types, there are hundreds of different strains. This is why you can get flu repeatedly in a single year or over the course of your lifetime, because you're not getting the same flu time and time again. There's variety. Now, you've lived in a time where we've had a few flu epidemics and outbreaks, and so you may be familiar with the nomenclature, the naming of flu strains. We say things are the H1N1 flu or the H2N5 flu. What we're doing is we're describing the capsid of the flu. We're basically describing it like we would a person, brown hair, blue eyes. H stands for a capsid protein called hemagglutinin. There are 16 types, so you could have an H1 or an H16 or an H7 in between. The hemagglutinin is essential for the influenza virus to bind to the host's lung epithelial cells. The N stands for neuraminidase. This is another capsid protein, and the neuraminidase is most important for viral exiting. It severs the tie with the virus particle, so the virus, once it's been duplicated, can go on to infect someone else. There are 9 types of neuraminidase, so you can have an N9, an N1, or an N3. And so when we say something is an H1N1 flu, we're saying this flu has the H1 shape and the N1 shape. Okay, we're describing it like blonde hair and blue eyes or brown hair and green eyes to describe what the outside looks like. It does not speak to the inside genetics, but just the visual external morphology. What makes flu really unique and dangerous in terms of a human health concern is the fact that unlike other viruses that have a single genome, the flu genome is segmented. It's fragments, bits and pieces of individual genes floating around. But because they're not inherited as a single circle of 8 bits of information, if a person were ever infected with 2 strains of flu at the same time, within that cell, these bits of information could be reassorted. They could be mixed and matched, swapped out like trading cards. Essentially, The flu virus is the only virus capable of having sex and having genetic recombination. And that is why there are so many strains of flu. That is why we haven't cured flu despite having a vaccine, because it out-evolves us. Every year, we are combating a brand new type of flu, the product of reassortment. So where do we draw the line between epidemic and pandemic? An epidemic, epi, means on top of. So this means that there are more cases than usual at this time. Seasonal flu, the type of flu that you get December, January, February, this is an epidemic of flu because flu is around all year. But during the winter months, that's when we see more cases than usual. Seasonal flu is the result of something called antigenic drift. Know this pairing, seasonal and antigenic drift. Antigenic drift is the result of subtle mutations, while the H and N stay the same. So while there's small changes in the genome inside the viral particle, outwardly it looks the same. So our old immune system memory responses to either having had the flu before or having gotten the flu vaccine is able to provide some protection. This drift comes from point mutations when the virus has been copied. Another way you can get epidemic flu is the result of an immune pressure. This is an unfortunate consequence of selecting for the one thing you have no protection against. Let's see this in a scenario. Assume our flu vaccine this year protects against yellow, green, and blue flu. But at a party, you were exposed to blue, green, and brown flu. In the picture here, we can see antibodies, the Y shapes, neutralizing the blue and green flus. But this leaves that brown flu unrecognized. And so that's the one flu that infects you, the one flu that is replicated, and the one flu that is passed on. Immune pressure, this selection for the one thing we did not anticipate or protect against, accounts for 20% of all flu cases annually in just the United States. Now, what makes a pandemic occur? These are sudden and complete changes, dramatic differences. This is usually due to the H or the N, the hemagglutinin or the neuraminidase receptors on the capsid being completely swapped out. So the virus looks different. Even if the genetic information inside is the same, outwardly, they're very distinct, and the immune system will not recognize it. Antigenic shift typically comes from interspecies transmission. I mentioned that most viruses have a narrow host range. Flu is an exception to this. There are flus for ferrets and pigs and beluga whales. And while humans cannot get the flu from these different sources, aquatic birds can, so geese and ducks, swans, and they become mixing vessels to potentially accumulate these different H's, N's, genes where recombination can take hold, and then they can potentially transmit it back to humans. It creates a very long 6 degrees of separation life cycle, but it's only a matter of time before differences in horse flu, chicken flu, dog flu eventually make their way into the human experience. Another way we can get antigenic shift, and I've already kind of alluded to this, is just recombination. If a single person gets exposed to 2 flus, becomes infected with 2 flus, because of the factorial expression of 8 different genes, that would give rise to 254 brand new combinations, not including the standard blue or brown flu depicted here. So this is one of those examples where if you're not part of the solution, if you're not getting vaccinated and preventing yourself from getting sick, you are potentially part of the problem, because in you, new strains of flu are made, and this in return gets passed on to the world. The last way we can see the pandemics reemerge is through the reappearance of a previous strain. In this case, it's the exact same strain that we saw previously, but enough time has happened that the population has no immunological memory against it. It appears as a brand spanking new, very dramatic difference. The picture that we have here, we can see the 1918 H1N1 flu hit, and we see a large peak showing it's a pandemic proportion, apocalyptic scenario. After that, we see small peaks, small subtle changes that represent the seasonal drift. But then in 1943, boom, we see the same 1918 flu and the same high peak of a pandemic. This was real life. And what was surprising was the fact that when the Spanish flu hit, the people who were dying were the normal 20-year-olds, 30-year-olds, people who we consider having good, robust immune systems. The 70 and 80-year-olds were not getting sick at all. And this is what led scientists to explore the hypothesis that it was the reappearance of a previous strain, and the elderly had been protected against the fact that they had already experienced it and lived through it in their 20s, and therefore had memory on their side. But the current 20-year-olds, when the Spanish flu came out, were experiencing it for the first time, and therefore took their chances. So some contributing factors. In terms of interspecies transmission, this can be sped up by very specific species. The aquatic birds and pigs are of most significant importance to humans. That's because aquatic birds can have access and express all 16 H's, all 9 N's, and they don't get sick like we see them get sick. They are infected, but they don't exhibit pathogenesis. You're not going to see a goose on the side of the road with a fever sneezing, and yet they shed virus particles left and right. They also travel. A lot of these birds have migratory patterns, and so they can bring genetics from Canada, Chile, France, to other parts of the world. Pigs are another concern, and that's because they are the middleman between humans and birds. Pigs have two size epithelial receptor cells in their body. So one is a perfect match for the same size as birds, and the other is a perfect match for humans. And so pigs typically get flu from other pigs, aquatic birds, and humans. And this allows them to also be mixing vessels, whereupon new combinations can arise. Now let's talk about prions. Prions are infectious proteins. Now remember, they're made up of amino acids. These are things that we eat. In fact, there are 23 essential amino acids that we are required to have in our bodies at all times, otherwise things fail to work. A person who's infected with a prion disease is not going to get a fever and immune cells directly out the gate addressing this issue. What makes the prions dangerous is that they are misfolded and damaged proteins that make them very hard to break down and recycle, very resistant in that sense. And because they're so large and clunky, they're tying up resources and potentially blocking blood flow. And this can cause areas of the brain to die. And as the brain dies, it will shrink. And so the symptoms of a prion disorder generally tend to look like a neurodegenerative disease because we are breaking down the brain tissue. And the disease, depending upon where those prions are located, can manifest differently. So if your motor sensors are what's breaking down and dying, then you'll exhibit lack of coordination. If it's your language centers, then the person, as they lose cell tissue, are going to exhibit slurred speech. If it's your mood centers and regulatory abilities, your autonomic responses and reactions, the organisms can exhibit varied mood swings. Right? And we are not the only organism that has prion disorders. We actually see prion disorders in cattle, sheep, and a number of ungulates like moose, deer, and elk. In cows, we call it mad cow disease, and that's because the predominant symptom is increased anger and aggression. In sheep, the prions are located in the touch sensors of the sheep's brain, and so what happens to them is as those sensors die, they feel like they're being touched and scratched at all times. And so the disease is called scrapie because these sheep will scratch themselves on fence posts and walls to the point where they create large gashes that can become infected. In terms of the deer and elk, what happens is it affects their energy centers and their ability to sense time. And so what we see is these animals become very lethargic, chronic wasting disease. They just unstartled, unwilling to respond to predators. They just kind of languish around. There's no real drive to eat or do things that you would see in a normal animal. In terms of people, the symptoms tend to look a lot like Alzheimer's: lack of coordination, slurred speech, mood swings, inability to retain information, memory lapses. And it's all because centers of the brain are slowly dying off and the brain is getting progressively smaller. How do you get prions? Well, it's important to point out that this is a very specific protein. The prion protein is already present in our brains. You and I both have them, okay? And the prion itself is not the problem. It is an abnormally folded prion that is the problem. So it's the accumulation of the PRP, the prion protein, SC, because it was described in scrapie first, that leads to disease. How do you get this abnormal prion? Well, sometimes it's just the result of spontaneous mutation and it misfolded incorrectly on its own. Okay, having one of these happen in your lifetime, generally not a problem. You could also obtain an abnormal prion because you inherited a disorder that makes you more likely or predisposed to spontaneous misfolding, in which case it's not just one in your lifetime, but several. But again, not very likely to cause disease because there's a number of normal prions in the brain as well. An example of this is Creutzfeldt-Jakob disorder, which is largely restricted to the Mediterranean Jew population. In terms of human disease, where we see prion disease occur most frequently is when it's acquired through food. Okay? And this ends up connecting to and speaking to a very unique attribute of prions. It's how they replicate, how they make more. But we'll talk about that in a few slides. Here, I wanted to address the difference between a good prion and a bad prion. The good prion, PRP by itself, prion protein in green, is a three-dimensional prion. It's got three-dimensional shape. It's got form and functionality. The abnormal prion, on the other hand, is composed of only beta-pleated sheets instead of beta-pleated sheets and alpha helices. And so this leads it to be folded back and forth, kind of like a flat fan, and it's only two-dimensional in structure. This makes it not have functionality, but more importantly, it makes it harder for the body to recognize and break it down to recycle those resources. A classic example of the connection between prion disorder and acquiring through food is the link to cannibalism. So the Donners, Serving Your Fellow Man from 1846, were a group of colonialists that were traversing from the East Coast to the West Coast in November. There were about 90 American pioneers making this move. They left Missouri and headed for California. When they arrived, less than half had made it. What happened was they were trapped in the Sierra Nevada for 3 months. Okay, this was a trip that should have taken about 3 weeks by cart and buggy, oxen. 3 rescue attempts were made, 2 from one side, 1 from the other, but they couldn't get through the passes. And so this group of colonialists ate through their grain stores, ate through all their food and resources, the leather binding on their books, trying to stay alive. They weighed pros and cons heavily. Do I need 2 ox to pull this cart, or could I get away with just 1? All right, we're eating one of those ox. Oh, Fido just died. Well, I'm hungry. Let's take care of that. They didn't turn on each other, but it came out much, much later on the deathbed of some of these surviving members that when a person died of starvation or from the elements, they made use of that human meat. And what we've seen is it's in these instances of cannibalism where you can inherit someone else's prions and absorb them into your own, and they can compound the problem very rapidly. It's because prions reproduce by a essentially peer pressure type mechanism. So in this story here, I've got a good 3-dimensional prion in blue and a bad flat beta-pleated sheet 2-dimensional prion in red. We have lots of the blue ones in our bodies. But let's say you accumulate a bad prion from a bad burger or because you ate Todd. The bad prion attaches to the good prion and it forces the good prion to change its shape and structure and conform to the bad prion, the beta-pleated sheet, the 2-dimensional structure format, converting from good to bad. And now you've got 2 that are dysfunctional, tying up resources, and harder to recycle. And that when they bind to another good prion, they forcibly convert the good prions you currently have into bad prions. It is the proverbial one bad apple spoils the bunch. And therein lies the problem with cannibalism. If you inherit 10 prions in one go, 20 prions in one go, 100 prions in one go, how much more rapidly can they convert the good prions that exist into these bad prion scenarios?